Okay, so I am a postdoc. I just recently joined MIGEC in Montpellier, IRD. And uh, for me, the main motivation is using phylodynamics or related methods to understand the ecological and evolutionary interactions in viruses at the animal and human interface, and using that knowledge to solve problems of epidemiological problems, and then feedback learning more about the ecology and evolution. One important aspect, uh, see, one important aspect of um, ecology is space, and I'll be talking about a work that I did in ENVT Toulouse with Timothy and Mathilde, Timothy Wen and Mathilde Paul, as a postdoc, where I looked at the spatial spread of HPAI, highly pathogenic avian influenza, a particular kind of virus H5N8, its epidemic, and evaluating control measures. This is essentially what Simo gave us a presentation about, is looking at phylogeography, like, how you understand how a virus spreads, right? And this is particularly important. Of course, this is important for any virus, but this is particularly important for livestock, because unlike humans or wild animals, livestock, their mobility, their movement is highly controlled by transportations. So it's very common, it's almost default that whenever you have virus outbreak, the first thing we think about, okay, how did the virus spread? Must be how the animals are moved between farms. And that is something we tried to, and people have been using a lot of different kind of modeling frameworks, like from network to agent-based models, but we use very simple modeling framework, something again very similar to what Simo talked about, but in a different phylogenetic framework called coalescent, more population genetic framework. But before that, let's briefly talk about the epidemic. To put things into perspective, this is H5N8 epidemic, which is, I think, happening since 2014-15. But the first big one happened in 2016-17 all over the Europe, but France was particularly hit badly by that. If you can focus for the first time, like first on this side, so this is where all these different colored dots that you see is the epidemic, the outbreaks happened in different forms. But as you can see, mostly it was concentrated in the southwestern part of France. And it is no surprise, because these viruses, as Claire talked about, they come from wild birds to domestic or poultry birds. And ducks are particularly vulnerable to them. And this is the area where we get the magredo canard that we are going to have tonight. The canard industry is here. It's very highly dense, and usually this part get hit hard. Although later on, we had two other outbreaks. The recent one actually this year, where you have actually spread outside of this area as well, but that's a story for another day. And this is the equivalent of from actually Claire's on the first paper on the outbreak, where she shows plots the number of outbreaks from left to right on the basis of beginning, which was like end of November in 2016. And then immediately there was, initially it took some time, there was some control measures. There are mainly two major control measures that we looked into. And by around the third week of March in 2017, it went over. Now, as I was talking about movement of livestock, that's important. And before I tell you how we put that into, we integrate it into our model, let's first see how that movement actually happens and why it is important. For the duck farm industry, it has two sets of farms. You have the breeding farms where the chicks are brought in, and they're usually outside, they're bred, and then after a certain age, trucks bring these chicks into these fattening farms, and this is where they are fattened for the foie gras industry. And the main movement of these ducks in their lifetime is from the duck breeding farms to fattening farms and then to abattoir for slaughter, right? So here the ducks stay a lot outside. So this is usually where you likely to get an infection from the wild bird. And as soon as there is an infection, it is detected, for example, here, this one, they stop any truck movement from this farm to wherever they are going, right? Similarly, when they detect infection in one of the fattening farm, so any trucks that it's receiving, they stop the movement of that. This is important to understand that in contrast, not all the truck movements are stopped in a particular region. Only specific truck movements are stopped, right? And Claire showed using networks what you see here in this paper that they published in 2020, that you have these lines are truck movements, the data between all different, I think all of these spots are different forms. And what she found, she estimated a particular risk based on a set of parameters of connectivity within a particular time period of infection, that these are the at risk, these few. So overall, what she found is that uh, there was not many long jumps of the virus based on the live duck transport. Rather, most of the transmission that was that was impactful were local transmission. So this is important to understand the context, and this is what we wanted to understand using genomic data. That what does the genomic data tells us about this pattern, and can we actually understand uh, the transmission? Is it also local, or there are long jumps or something like that? But before we go there, the second really big control measure that happens is preventive culling. Uh, just to explain briefly that what does it mean. So you have culling and then you have a preventive culling. Culling is when you have an infection in a farm, you clean all the animals, you kill all the animals. That is culling. But then in this industry, and I suppose many other livestock industry, another preventive, like one step preemptive rather, measure is that if you have an infection in one particular farm, you preventively cull all the other animals in all the other farms within a certain distance. And there are logic and arguments and counter arguments, how far you should cull and all those things. Uh, but what a paper that looked into the epidemiological data that they found that they found what mattered is the delay in culling, like when the detection and the culling, but there's not much impact of the delay between the detection and the preventive culling. So there was a lot of question about to understand more detail that how preventive culling is useful, because this is also an ethical question and an important public question, because you are essentially killing a lot of undetected, uninfected animals. Is it worth it, right? Uh, so on the basis of that background, just to give you an idea how the preventive culling happened. So initially the infection started somewhere here. This is the tongue, this the Debapriyo, and by that time, within I think 2 weeks, it already spread here, which is Jets, and then this is Long. These were the two departments which were most infected by that time. There was the infection, like infections already here. The department started culling here. It's like a barrier to a wildfire. You preventively cull all the animals, and you expect that the fire will not spill over to the new other area, right? So they did the culling in three phases. You had the first started in 4th of January, still spread. So they increased the area of the preventive culling on 14th February, and finally by 21st February, they just culled like around 2,000 or so farms all over this area. And by over like March, the epidemic died down. So does the preventive culling actually have any useful impact? That was another question that we had in mind to test with viral genomic data. So the modeling framework that we had in mind is a simple a model where we treat the patches, the populations as patches, and the data that we had from the Southwest, the study was focused on Southwest because that's where we had the most number of information and data. So we had mainly data from the 7 departments. We assumed very basically these two assumptions is that the populations are like patches, and then you have a movement of livestock between them, and then there are movement inside them. And based on these two assumptions, we are going to measure the migration of the virus. And I'll tell you why I call it a migration. It's a bit strange to hear that virus is migrating, but it comes from a genetic framework called coalescent, right? But anyway, so before going there, so essentially this was our framework of understanding how the virus spread between the bacterium. Now, I use this tool called MASCOT. I heard this term in between talk discussion, and this was developed by Nicola Muela, who used to be a PhD student with Tanya Stadler, now in Seattle. And this model actually links the spatial model to the phylogenies in the sense that it uses, so there is this method called reconstruction of ancestral state, where you try to understand, just to give you an idea, this is from Nicola's paper in 2017. If you have these two sets of populations of different color, how do you understand where the root of the phylogenetic tree is located? And what has been already established that probabilistically you can say like what is the probability of the root to be in one population versus the other population. What Nicola did, he developed an algorithm to make it computationally much more feasible, much faster. And here, what you see essentially, so MASCOT is the first name for MASCOT, is a green, that the estimations are pretty good in compare as with, I mean, he compared with all these different sets of methods. The second interesting and useful aspect of MASCOT, and particularly I want to mention because we had a discussion about data integration, is that often information in case of epidemics are scarce. And one of the goals for us to how we use scarce data and then use other kinds of data to make sense of the actual epidemiological data, right? So, I mean, again, Simon made it much easier for me. You already know what is that, the data integration. So you pile up different sources of data together in a particular framework to make sense of it. This particular method that MASCOT uses is originally came from actually Philippe Limet's group, but in a different version, in a different flavor. What Nicola did, he allowed estimation the change of this migration rate and the effective population size, that is any over time. Over time. And the framework is a GLM framework where you simultaneously estimate this viral migration and effective population size for a range of covariates. Each of these covariates are different sources of data you get it from. Um, so basically, this is the methodological part, the framework, and we, oopsie, right. And so we had used a large number, like not a large, not like in the SARS-CoV-2, this is nothing region, but we did have genomic data, one genome sequence from each farm. We had a 196 sequence, and we had and we got it from our collaborators in ANSYS, Plufagan, the virology lab, and then Claire very kindly provided the livestock movement data, the epi and the culling data that we had, and uh, that is the data part of our work. So there are a lot of things that you do, but broadly you have the descriptive part where you reconstruct the viral spread, and then you have this predictor analysis where you have these different kind of covariates, and you try to see how these different covariates actually inform both the migration rate of viral spread and any of the virus. And this tells you, anyway, I'll come to that later. First, let's see the viral spread, the phylogenetic tree. And this is the MCC, the average tree of the viral spread. And what you see here, the x-axis, you have the spread of the epidemic. So from left to right, the epidemic spreading. And the colors tell you are corresponding to each of these patches of the Debapriyo. And when you see a change in color, that tells you that the virus spread from one Debapriyo to another Debapriyo. So for example, if you have this change here, so this is this Debapriyo, I think this is Oppidinis, to this other Debapriyo, which is Lond here. Yes, and so that tells you broadly that how the virus spread between patches or between locations. So one of the conclusions from this figure, what you see, if you track the change in color, the change in color are between adjacent patches. There is most of them, there is no long jump. For example, there's not long jump between Than to Lond, or from Than to Oppinis, like that. So this kind of tells us again that the virus possibly spread in shorter distance, not in long jumps. Another thing we can do just to understand that if our model is giving us a proper estimate of any. So one of the things we did is looked at any, for each patch has separate any, the effective population size, and we tried to understand if it actually tracks the size of the epidemic or the case counts. I just show one of the Debapriyo, which has the most amount of data. So what you see, these bars from left to right are the case counts, and this red line is the estimated NE by MASCOT, and it's more or less, it tracks the number of cases for the Debapriyo alone. Now the covariate analysis, the GLM analysis, and first I'm going to show you the analysis for the viral spread, and then I'll show you the analysis for the viral any. The analysis for the viral spread, it essentially uses the movement of duck consignments between the Debapriyo and tries to understand that if any of the covariates of such explains the spread of the virus. On the other hand, the analysis of the viral any is for within each patch. So it is trying to understand the size of any or the value of any of each within. Does it vary or it is affected by any of the covariates? If it does, it gives us an indication that there's possibly some kind of movement happening within a patch or it is important for the viral spread within patch, in contrast to between patch. So first the viral spread, and what we have here on the left side, you have a range of covariates that are important. We thought important, but I'm going to, for the time being, only talk about those which we found are impactful. If you have any questions later about the other ones, why we found, why did we incorporate them, I'll be happy to answer. The first thing, and the impact is measured using Bayes factor. The more the value of the Bayes factor, the more impactful the effect of that covariate. So the first covariate that became most impactful is what we named here as 4th of January, which you might remember was the first time the culling was initiated. And what it essentially measures the migration rate before culling versus after culling. And it says that the migration rate actually changed, it actually reduced after 4th of January culling began. What is interesting is that, okay, I'll come to the interesting part later. It also found another thing that was impactful was the shared border. Essentially what we said that see if the patches of the department with shared border, if that has any effect, and says that yes, patches which actually had shared borders had more impact. This is actually a confirmation of what you already see in the phylogenetic tree, but we see that the viruses are moving between adjacent patches. What these covariate tests is that the effect of number of consignments between department, the duck, live duck, consignments between department, if it had any impact. It didn't, which tells us that although the ducks moved between shared borders, between patches which are adjacent, but that was possibly not because of the number of consignments they exchanged, the live duck transport. The interesting things I wanted to just point out is that while we had an impact of the first culling measure, the other culling measures on 14th of February and 21st of February didn't seem to have any major impact. Now, looking at the NE, the effect of the covariates on NE, and of course, what we find is a very strong effect of the daily case number that I already have shown for long, that it tracks it strongly. The second covariate that we found is movement within the department. So these are consignments of live ducks that were exchanged within department, not between department, and they seem to have a marginal effect on the NE. Surprisingly, what we didn't find is that the density of duck farm, which we would expect would have a very high impact, because you would expect areas which has much higher density of ducks will have much more infections. We didn't really find much effect of that in this model.
Question? Sorry.
Yes, go ahead.
Were you testing those predictors one at a time or all together?
All together.
All together.
Yeah.
So the daily case numbers correlate with it extremely well.
Yeah.
So then that doesn't really give you much of a chance for any of the causal things to happen.
Yeah, that's true. Yeah. So some of the, so we did look for correlation, but what is interesting is that if I remember correctly, the density of duck farm and the daily case number. So one of the things it also does, I think, if I remember correctly, the MASCOT, it has this inbuilt indicator which tests for which one is more important. So I did run without, for example, these density duck forms and all these different. So I think MASCOT takes each of them, test it, and then add the next one based on the effect of the other. Isn't it?
The indicator you have
The
is
indicator.
impacted by the indicator you have for the others. So I'm not sure there is an order of adding them.
Right. There is an order.
But there is an impact, as you mentioned, and if they are highly correlated, they will negatively impact each other in terms of indicators and then base factor estimation. But it's also a piece of information.
Right.
So
exactly.
if you have highly correlated predictors, that might be good to indeed redo an analysis where you only select from.
But I remember that I removed the daily case number and density of duck forms. It didn't really show any impact.
Are they correlated when you look at them outside of the analysis?
I did. I think some of them, I think the movement, I clearly remember that the movement and the density of duck forms are correlated. And another thing, yes, I think daily case number and density duck forms are correlated. But another important thing is that I think the case number, which is the data itself for the model, is clearly driving the model a lot. It's actually a large part of the information of the model. So it's actually not a surprise that daily case number will have an impact. This is something I think you also discuss in that, not you, I mean, I think the Dudas paper, I think, the Ebola where they track, this is something similar that has been seen there, that when you use the epidemiological information as a prior, you actually influence that.
Yeah, it's a good thing.
Yeah. So broadly, the major takeaways are that preventive culling initiated on 4th of January reduced the viral spread between Debapriyo. Within Debapriyo, we had duck movement, which is weakly associated with any, which possibly indicates that some input of duck movement influencing the viral spread within Debapriyo. Virus spread across adjacent Debapriyo, but not by shipments to. So we don't exactly know how. There are some speculations, some hypotheses. One of such being as fomites. So some of those farms which are adjacent might exchange equipment, and they might actually carry. We don't know for sure. There is also one hypothesis that often what happens that trucks, they move, they enter, by the time they enter farms, they get a call that no, you should stop. They don't know, they don't stop, they move out. We don't know if that had any impact, if the virus gets, even the animals are not taken out, if the virus gets out, but the short answer is we're not sure. And as we just discussed, that number of infected farms is an important predictor of any. So so this is the work that we published this year. I did it in NVD. So now I'll just briefly talk about. So this is Benjamin. I'm working with him at IAGDE, and the projects that I'm looking at is in humans. So the idea is, for me, is I looked at some aspects of viruses in the animal side, now looking at some aspects of viral dynamics in humans. And here I'm focusing on interactions between the viruses, not the spatial aspect per se, but there's also some work I've done. So I've just started this work, and what I'm doing right now, I have the data, and I'm estimating epidemiological parameters based on models and also phylogenetics. And then I'm simulating based on those epidemiological model, both jointly epidemiological parameter as well as phylogenies, and then comparing to understand if a particular sets of epidemiological model are actually driving both the evolution and epidemiological parameters in turn, the interactions between ecology and evolution. I'm using master to do that. I'm doing mostly two projects. In the first project, using SARS-CoV-2 in Mexico, we have a collaboration, and we are getting both epidemiological data and genomic data, which are also publicly available. The main focus here is that, as you know, like everywhere else, there are a lot of different variants circulate. Actually, there's all of these, so many different variants are circulating. Of particular interest is this B11519, which was first reported in Mexico. So one of my goal is to understand how the interactions of this particular variant for the other different variants happening, and looking at possibly some test of selection and adaptation, but that's for next, possibly next year. The second thing I'm doing, actually, I'm using this as development of a framework to use the simulation-based master analysis. Is an interesting little epidemic that happened a few years ago in Cambodia, where we had a small village which had an epidemic of HIV. And HIV, of course, spreads sexually, but it was found out that there was a quack, a doctor who's not trained, was actually using syringes that are infected with HIV. And people found out, and then they did surveillance, and there was investigation, and found out that that seems to be a huge, huge impact. So one of the things so we want to so you have the susceptible and you have the HIV, and when people are investigating, it was found out some of those samples which are HIV also has HCV. And so it became clear that HCV, parallelly there is an in parallel epidemic of HCV going on. So I'm trying to understand the contributions of both the sexual transmission as well as the nosocomial transmission, that is to the syringes, and if we can understand that the true size of the HCV. That's all. I'll stop here, and thank you very much. And thank you, Julia, for you and the organizers, Gail, Samuel, everybody, Patrick, for giving me this opportunity. And thank you for your patience. Thank you. Yes.
Regarding the first part of the presentation, so you performed an analysis at the level of departments, but could it be also possible to consider an investigation at the level of the farms? So if you get a phylogenetic tree connecting all the infectious cases, so all the farms, could you try to use that tree to relate the evolutionary relationships between the cases and some connections with, for instance, you know that some trucks are involved in the same network connecting those farms, and do you see a cluster in the tree with those kind of external data connecting the farms by the same truck company or familial relationships, not with necessarily an ancestral state reconstruction, but just by looking at the phylogenetic signal that you have. So if you have the conservation of that trait along the tree, for instance, the fact to belong or to use the same truck company, that could explain the clusters you observe, but it's another question, because the predictors you tested are at the level of the entire epidemic and between departments. But if you want to connect the farms.
Very much, yeah, it's a very pertinent question. I had a brief discussion with team about it. We did actually try to, and Claire, possibly she has much better idea about the truck movement data. number one, what we were discouraged by was the truck movement data. We didn't have like the complete set of, so as she explained that often you have cooperatives and like she explained in China, but even in France, there are five, I think, 4 or 5 truck companies which move between different regions and different farm, and the data that we had is incomplete. It was
So
not
just for one of the companies or something like that?
Yeah, only 1 or 2 of them. So we don't have and also not only that, we only had data not from the full phase of the epidemic, only had a part of the epidemic. Another PhD student in the same department who only looked at network of truck movement. I think Billy was ill. He looked at and yeah, so he looked at that. And what interesting thing was that he also found, just to bring it back with the density of farm, that it seems like the density of farm thing is complicated. It seems, again, correct me if I'm wrong, Claire, that what he found is that there is not a straightforward direct relationship, right? That the movement and the spread of the virus was not equally straightforward. Yeah.
Like movement in different ways. It seems like it's not really like at least the movement of live was not really influencing spread.
But again,
It's
at the
more
level of the
something
departments.
No, for him, farm.
No.
Ah,
without,
So he did
okay,
networking on the farm
yeah.
networks.
So it seems to be something more linked to the density of farm.
And did you also have some assumptions about the impact of wind? Because I'm asking this because I know that for even influenza, it's a theory remaining. In Belgium, we tested that with something, with a new approach, and we did not really find a correlation between the direction of the spread and wind. But I know it's something coming back on the table again and again.
Yeah, we tested, but it was just a specific case. So we tested something in one valley of Pyrenees. And basically, we worked with Météo France, because the idea was to use one of their models, because they developed model to test, like, to identify areas at risk of having, let's say, there is an industry having an issue, and then to estimate where the different chemicals would go. So we use the same model, but for the source would be a farm instead of an industry, and we would say, okay, like the wind will spread like the ice particles. And so, but this was just for a specific case, and actually, like the direction of the wind and the direction of the outbreaks were completely opposite.
And so you compare the directions, not the intensity or the number of cases.
No, we look at the direction of the
new cases.
waves, like they predict like different waves over time, like over a few days.
Okay.
And we looked at where they were spreading.
But it was based on occurrence data, not phylogenetic information.
No.
Okay, so you're not sure that the new cases are actually connected to the previous one?
Yeah.
But now, I think from the last couple of outbreaks, now we have, like when I was there, we collected swabs from inside farms. So Claire will do that in the future. So now we can actually track.
She's on holidays.
That's right.
Yeah, so I think that's an open, so we discussed about that also briefly in our paper. That is an open hypothesis still, I think, worth looking into. The wind.
If you had the data for all the truck companies, something you could do is to look at the frequency of connections between farms, and reproject those farms in a new space, not defined by geographic distance, but defined by the degree of connections, using that network of trucks. And then you could look at all is the tree shape within that space. That
is one of the incomplete, because it couldn't work. I did that actually. So I estimated genetic distance, and the truck company couldn't find any impact.
Okay.
So one of those projects. Unless, okay. Thank you.